ask them about their project involving how to analyze, how to interpret, how to integrate this data in this session right now. So please, Chris, take it away.
Thank you very much for the introduction. So yeah, as Robert said, my name is Chris. I previously did my PhD with Stein, and I now head the VIB Center for Brain and Disease Research Single Cell Bioinformatics Expertise Unit. Alongside that, I'm one of the lead developers of both Vision, which is one of the main pipelines used for the analysis in the FCA, as well as Maxime Douagnier and Chris Flerin, and I'm one of the lead developers of Scope alongside Maxime Douagnier as well. So with that, I'm going to start into the talk. So you're going to see quite a lot of overlap here with Hongji's talk, but that's actually quite good for the demos I'm going to show. So the focus on this talk is really about the data mining and analysis and the data visualization and query of the FCA data. It's a huge amount of data, and how are you going to be able to leverage that and look at that in your own analyses? So there's been an incredible amount of work, and on the data analysis side, we split this into kind of three main components that I'm showing here, each focusing on a really important attribute of a part of an atlas. So Vision, I mentioned previously, is a main analysis platform, and we're using the ScanPy package for the analysis there. And this covers automated analysis, the reproducibility of the analysis, as well as tracking of the data. We've got Scope, which I'll talk about in the second part of the talk, which is for data visualization, as well as the community annotation for this amount of data. There's a huge amount of cells across many different tissues, a lot of cell types to identify. And Scope was one of the main tools used for this during the Jamborees that you've heard about. And then ASAP, which is developed by the lab of Bart de Plancker, which is for more data visualization, but also fine-grained analysis. So it's another portal into the data. Alongside this, there's also ad hoc analysis to be done, for example, automated annotation and consensus annotation. That's not going to be touched here. So a very brief overview of the pipeline. It's not technical, just to give a very quick overview. We've tried as best as we can to follow publish best practices for analyzing the data. And this is a very broad overview of what it actually looks like for the analysis. You can see each of the different steps here. Importantly, we have batch correction in there because without that, we don't get such nice clustering. We have clustering, etc., with the Leiden algorithm. There's two really important parts of the pre-processing I am going to touch on briefly, just because they kind of set the stage for the types of data you are able to look at in Scope. The first of these is an index hopping filter. So we noticed pretty early on that there was some crossing over of reads between the different indexes of the different libraries that we have. And so on the left, you can see a heat map of the barcode overlap between a set of selected samples. It was between 1% and 5%, but that can really throw off. It can add cells to a data set where they're not supposed to be. And likely because a lot of the samples are pulled together on the same Nova 6. So to solve this, we actually use the 10x index hopping filter software to remove them. And on the right, you can see how that altered, and we remove this effect. The second one I'm just briefly going to touch on is the ambient RNA. So as well, we discovered kind of in the version 1 of the analysis that there was a set of what we were calling black hole cells. So they were expressing 3 different markers for very specific tissues. So here they were showing grainy head for epithelial cells, MHC for muscle cells, and 1-cut for neurons. And you can see right in the center circled on that left plot is a bunch of cells which are expressing all 3 of these markers. So we actually tried a couple of different methods, supex and decontext, to remove these effect, and we settled on decontext, which was built into Vision and was used for the rest of the analysis. So that left us with these 2 datasets, the V1 or raw dataset and the V2 stringent filtered dataset. And the difference there is the boxes in orange. So given that's what the data actually went through before the pre-processing, where are we now? Now, this is the slide that was just shown by Hongji, so I'm not going to go into it too much, but we have 17 tissues. We've got over 250 cell-typed annotations, all by many experts, primarily done using Scope. And in the right, you can see this is the t-SNE of the entire dataset. This is all of the tissues integrated, colored by their tissue. So with Hongji's talk and with the few plots I've shown you here, you may be wondering, well, how am I going to be able to look at this data? How am I going to be able to see it in this way? Am I just going to have to look at the figures or else, or what? So that's where Scope is going to come in. Scope is going to remain one of the main portals to the Fleiss Atlas data. And although some of you are going to know Scope, some of you may not know Scope, and given that it's going to be a really key place to go look at this data, the rest of the talk is going to be a set of demos to show you how you can use it, how you can leverage some of the functions to investigate the data. A couple of caveats here. One is these are all pre-recorded videos. Hopefully they'll play at full frame rate, so you'll see them nicely. We're at 500,000 cells, which is the highest we really used with Scope, and that can sometimes bring some kind of long waits. So all of these videos are edited to make the pauses go away, the loading times are shrunk, but you'll see everything that you can do. You'll just have to wait a bit longer if you do it live. So to start with, if you go to Scope, you're going to see this home page. On the left, you can see here we've got a different hierarchy of the different files. You can see, as I mentioned, we've got the raw and the filtered data sets, and we've got them for the all integrated analysis, as well as all the tissues. You've got a few buttons there where you can download the data and things like that. And then, like I mentioned, for the different tissues, you can see the V1 and the V2 data, depending on what you want to see. You can just click on a data set to bring it up. It'll highlight to show that it's being brought up, and you'll get displayed with the plot. By default, we show the UMAP in many cases, but on the bottom left below the data sets, you've got a nice little settings bar which lets you switch between the different dimensionality reductions. Here, I'm going to switch it from the UMAP to the TSNE, and you'll be able to see a very familiar plot that we've seen already in a bunch of these slides. And there's some other settings here that you're able to leverage and play around with to change how you're looking at the data. The next... Come on. Nope. Let me switch out and reshare that. Demo videos were supposed to relieve the risk of doing live demos, not make things worse. So the second demo I've got here is searching for genes. You've picked a data set, you're looking at your data set. Where do you, how do I look at a specific set of genes now? So in Scope at the top, you've got multiple, you're now seeing your TC. At the top, you've got multiple search bars here. This lets you actually see different genes in different colors. So here I'm going to search for grainy head, like in the previous plot. In Drosophila datasets, you get to see the different synonyms, as well as if this gene is a marker of one of your clusters. I select it, and you can see that we've shown up the cells with the grainy head expression in red. You'll also notice the little sidebar there that lets you change the scale. I can then do the same for a couple of other genes, MHC here for the myosin heavy chain to highlight the muscle cells, and they highlight up nicely in the top in green, and then finally one cut for the neuronal cells, which is going to show up in blue. What you'll notice is that you've got a couple of cells here that are highlighting in different colors, in purple and in yellow. And this is much like in immunohistochemistry. You're going to get the combination of the three primary colors here to be able to see where genes are expressed, multiple genes are expressed in the same cell. It's of course going to do this again. So we're going to drop out and drop in. This is going to be a little bit annoying. I apologize for that. The next part I'll show you is looking for an annotation. So I showed you the genes there. You can also look for a different annotation. The Fleiss Atlas has many different tissues. And so the tissue, for example, you can search for tissue, or you can search for one of the categories of tissue. So gut here I've selected instead. And that lets you bring up the, by searching gut, you can bring up the tissue, and now you'll get the overview of the annotation. This is the plot I showed before. On the right, you've got the legend there with the different colors, and on the top of the plot, you've got the different labels for the different annotations you've got there. And these annotations could be a variety of things. We've got many things in here, including the batches, etc. For the on top of the annotations, we've also got clusters. So with the clusters, you can search for this by name. So for example, indirect flight muscle, I'm going to show and use this as an example throughout. You can also search by a cluster number, however it's been named in the file. This highlights in red as a solid color. It's either true or false that it's in this cluster. But on the right here, this is how you'll see the annotations that were made during the Jamborees. You can see it was made by the FCA BioHub and with 6 endorsements from different people. Below this, you've got this marker table, and this will show you the differentially expressed genes in this cluster versus the rest of the data set. And so here we're looking at the indirect flight muscle. You can see the top 2 highest differentially expressed genes with log fold change here are bent and flightin, both very known for their role in muscle. And this is against the rest of the cells, like I mentioned. Clicking on the more info in the annotations will let you see any comments or markers that the people that annotated this have left. And for the data coming from the FCA, we've left specific information in there. And then finally, underneath the marker table, you've got 3 different buttons here that are going to do some pretty useful functions. The first of these is gprofiler. So gprofiler is a web tool developed by, not by us, but it's a web tool that lets you give in a set of genes and look at the gene ontology enrichment for those genes. So we let you select however many genes you want to show, to take with you, how you want to sort it, and which species you want to put in. We'll send it directly to gprofiler, and you'll be able to get pretty quick results out of the set of genes. So here you can see contractile fiber and microfibril assembly, which makes a lot of sense considering we're looking at the markers for for a set of muscle cells. You've also got the ability to download the marker genes from that cluster from that table as a file, and finally to download that cluster as a separate Loom file, which you could take and use in your own analysis elsewhere. Next, you can look at, instead of a single cluster, you can look at all clusters. So any clustering in your Loom file, you're able to search for all clusters, and this will let you see a complete overview of the entire clustering. This looks a lot tidier than the annotation because it's based on the neighborhood embedding, and here you can see the example I just showed of the indirect flight muscle, as well as all of the other different clusters here. By default, this would show the cluster name that's in the Loom file that you gave it, but in place of that, if annotation was performed on scope, if there's a annotation with a positive amount of votes, then we'll actually bring that up and we will replace the name with that so that you can annotate this on the fly with multiple people, and the new annotations will show up as and when they're made. One of the other really useful parts of scope is using the Lasso tool. So on the left, you'll see that we've got 2 tools here. One is just for moving the plot, and the other one is for selecting certain cells. I'm going to stick with the same example of the indirect flight muscle. It's a big data set, so it's a bit slow, but you can select these cells, and you'll see they highlight up in a nice color to let you know you've selected them. On the right then, under the Cell Selections tab, you'll see the selection, which shows you how many cells you've got, as well as which clusters these cells are within. So we show the overlap with all clusters from the dataset. So you can see different clusters, the clustering they're from. You can make the table a bit bigger, and you can see in this example, I've highlighted the indirect flight muscle cells. It shows that I selected just under 9,200 of these cells. Of the 9,300 that I selected are in that cluster, and you get the percentages here. We've also very recently added the ability to download a Loom file of the selection of cells you made. That means you can select any particular set of cells that you want and download them as a Loom file, which you can take on for your own further analysis. One really interesting part from this is that Vision, the pipeline that we've used for analyzing the Fly Cell Atlas data, will directly take in these Loom files and will let you analyze them further with any of the algorithms we have in there, which does include Harmony for redoing the batch correction. The thing that's really important to know here is when you download that Loom file, you get the raw gene expression matrix. You don't get the normalized one, which means if you don't do a batch effect correction, you're going to have batch effects between the tissues and samples. So Hongji mentioned the annotation that we've done, and I've shown it a few times here, and it's important to know that we've actually got 3 forms of this annotation. The first one is just labeled in these datasets as annotation, and this is using the jamborees. This is going through all of the different jamborees from the different tissues, combining all of them together and coming up with an annotation for each of the cells based on how many votes each cell got, and this is across multiple different resolutions of clustering and all the different datasets. And as well as all of the machine learning, you can see it's a very busy plot. This is actually a beautiful plot to scroll around and see how the groups of different cell types combine together. Again, the example I used previously, you've got the indirect flight muscle up there, and you can see all of the different muscle types that are together there. If you keep searching for annotation, you've also got broad annotation and broad annotation extrapolated. So these are basically, we've taken the narrow annotations and we've grouped them by the broader annotations extracted from gene ontology. So now we just have muscle system rather than the specific ones. And you can see that there's some cells spotted between there. And then if we go to the broad annotation extrapolated, this is where the all data set has actually been performed a high resolution clustering at 8.0, and we have assigned all cells from each cluster to the most represented, i.e. more than 20% broad annotation. Some manual annotation was done in case a cluster didn't pass a certain criteria, but this gives you a broader and, as I mentioned, extrapolated analysis. And you can see some of these smaller clusters with different colors around the muscle systems. Then, one of the other really useful parts of Scope is the Compare tab. So this is actually coming back to a bit about Erica's question in Jay's talk and what Hongji showed. You've got on the left here, these are all the, again, I'm using the word annotation here. These are actually cell annotations, i.e. the tissue where the cell came from, or the sex that the cell came from. So which sex was the fly it came from, the batch, this kind of thing, the age of the cells. These are all here. And on the Compare tab, you can actually select one of these and drag and drop the different categories onto different viewers. So here I've split the cells between the male and the female cells, and this looks identical to what Hongji showed. And doing this, and I'm going to show exactly the same example, you can search for ROX1, ROX2, for example, and you'll see that in the male cells, ROX1 is very heavily expressed compared to in females it is not. In the top right there, you get this nice little box plot that shows you the expression differences. And you'll notice even in male cells, there's some cells that don't express ROX1. Going back on what I just showed with the Lasso tool, you're able to kind of select these cells, see what it is, and you can actually say in this case it's the spermatocytes, where the ROX1 is not expressed. I do really apologize for having to switch back in and out of the presentation constantly. Another part of the data, which I haven't shown so far, I'm going to actually go to a different tissue here. So I'm going to switch over to the head data set, and we're going to look at Regulons. So this was shown earlier. Again, Anna introduced SCENIC a bit for me earlier, so I'm not going to go over what SCENIC does. But we're able to look at Regulons. So this is transcription factor networks within the data, and we've run SCENIC across the different tissues. In this case, we're looking at head. So I'm going to look at two particular Regulons here. The first of these is for glass, and those of you that know glass is a master regulator of the photoreceptors. You can see here that we're showing glass either as a gene when it's targeted by another transcription factor, or the top entry here is actually glass as the motif, i.e. the transcription factor and the Regulon for glass. And you can see here beautifully, we've got really two nice clusters in the top left here that have very high AUC values for the actual Regulon itself in the left plot. The top right plot shows kind of a thresholded version of the left plot. Just you can change the slider up the top there to change which cells you see pass it and don't. And in the bottom right, the expression levels of the transcription factor, in this case glass. You can also see the motif that was detected for this Regulon. And we ran SCENIC, actually it's a stochastic process, so we ran SCENIC over 100 times for these analyses, and you can see how many times a target was found to be targeted by that transcription factor across the 100 runs. So there's a lot of them that are found 100 of 100 runs here. I'm also going to show lozenge here. This is for the cone cells, and you can see we've got another different cluster down in the bottom right here that shows up. And here I'll show you where I changed the thresholding to limit which cells in that kind of thresholded plot that lets me see which are the ones with the highest AUC value, because before that there was a lot of cells which had some activity for that Regulon. You can again go through, use the Lasso tool, highlight them, find out which type of cell that you're actually looking at here. Both the cone cells and the photoreceptors you're able to find. So Hongji gave these showed a very quick overview of these really nice plots. Maybe you've got, the paper for the FCA is out, you've seen the whole head figure and you think it's really interesting, and there's this set of gray cells which are unannotated, and you might want to say, well, how can I go look at this more in detail? So if you were just to do this in one quick short video, you could go to the whole tier, select the whole tier dataset, switch it over to a t-SNE, and you go, show the coloring by annotation like was in the figure. You see the unannotated cells, and now you want to know where they are in the all dataset, for example. You can't just search the annotation in the all because they were unannotated. But what you can do, another part of the Compare tab, is you can actually use the multi-dataset function. So you'll notice I've switched over to the all dataset, the primary dataset I want to look at, and now I've used that little slider to select the drop-down to select the whole tier dataset. Switch it over to the t-SNE, and I know it was that small cluster of cells that I wanted to focus on. I can use the Lasso tool, and I can highlight these. And this is specifically because the cell IDs are the same between the two datasets. When I select them on the right, they'll actually show up in the plot on the left as well. So I can really narrow down exactly where those cells are in the full dataset. I can zoom in, I can just remove that view now that I know where I'm looking in the main data t-SNE. I can look at the annotation, and then I can say, well, I know they were unannotated, but what about the cells nearby them, for example? So I can make a selection, I can scroll through, and I can see, for example, epithelial cells or automagen cells that could be possible, possibly give me a lead onto going further with these unannotated cells that I might have an idea on where I can look at next. For the very last demo I have, I'm just going to very quickly show this is a different session now. This is the cross tissue analyses that Hongji mentioned. You're able to also find them. They'll be in the final data set available on Scope. And so this time we're looking at the epithelial cells. I can search for tissue, and I said these are cross tissue. So now you can see where each of these cells came from which tissue they came from. You can see that we've got some cells from the antenna, from the body, in the case of the body and the head. A lot of the cells, almost every cell that you find in a tissue, you find also in the body or the head data sets. So no more switching backwards and forwards. With that, the process data we have and the data availability, we've got the 10x results, the relaxed and stringent data sets. We've got the 10x cross-tissue results, and we've got the SmartSect, which I haven't gone into here, but you'll be able to explore this in Scope 2. These will all be available in both Loom files on Scope and in ASAP, and other platforms you'll be able to download H5AD files, for example. So after the data analysis, we went from vision, we went to these Loom files. You see these beautiful plots. You can visualize these in Scope. You can do the annotation, the marketings, the transcription factor networks. You'll be able to look at these in ASAP, which is what Vincent's going to talk about next. And you can put these back to Scope. What's important now as well is with this highlighting and selecting cells and then re-downloading a Loom file, if you wanted, you could really make a selection and put that back into Vision and go and do a further analysis of it on your own. So with that, I would have played this really amazing video by Jasper again. I will probably play it anyway, just because it's great to see all the credits of everyone. And while that's going, I can take any questions. I am realizing I can't see the videos of everyone here. Ah, there we go. I've got it. So if anyone puts their hand up, I couldn't see, but now I can. So I can see a couple of questions in the chat. Indeed, as Stein mentioned, the FCA session in Scope is not public yet, but it will come with the release of the paper. It will all be available. There'll be a link in the paper. You'll be able to come and do everything I just displayed there. And yeah, Robert, indeed, the compare tab really lets you do a lot in Scope. It really lets you explore the data in ways not many other, without going computationally in depth and running your own notebooks and things, lets you get quite deep into the data in different ways. Yep, the version 1 and version 2 will both be available in Scope. We'll keep both the raw and the stringent data. And of course, the raw data from the FCA will go up on like GEO, where you'll be able to download it and go right from the beginning if you wanted to as well.
I would suggest that, I'm sure there'll be some more questions coming in. I'd suggest that we'll switch over to the second presentation and then address questions in bulk.
Yeah, that sounds good.
Before the break. I mean, after this, we have a short break.
Yeah, so I'm still the host, so I'll introduce Vincent briefly. So Vincent is a senior staff scientist working as a bioinformatician in the lab of Bart de Planck at EPFL, and he's going to give a primer, but this time using ASAP instead of Scope.
Thank you, Chris. So I'll try to be not too long because I will probably go over the break. So to continue Chris' presentation, so what I wanted to present you is basically how you will be able to access